Welcome to video series on lipid metabolism. This video is what we'll learn in this video: oxidation of fatty acids in a simplified way, their clinical correlation, important MCQ discussion, probable university based questions, and memory tricks revisions. Now, why is this topic important? From exam point of view, these are the questions that will be asked. Describe beta oxidation of fatty acid, explain the energy yield, describe alpha oxidation, describe omega oxidation, describe oxidation of short chain fatty acids, explain oxidation of very long chain fatty acids, explain oxidation of unsaturated fatty acids. So each of this oxidation of fatty acid can be asked either as a 3 marker, 5 marker, or a long essay. And from the patient point of view, these are the disorders you need to remember. These include Ruxolic species, propionic acidemia, methylmalonic aciduria, medium chain acyl dehydrogenase. Let's understand beta oxidation. Beta of the beta carbon atom. The process takes place in mitochondria, and fatty acids up to 18 carbon are oxidized in the mitochondria by beta oxidation. And for long chain fatty acids, they are oxidized in peroxisomes. So there is a difference in activation. There is a difference in oxidation of short chain, medium chain, and long chain fatty acids. Short chain fatty acids up to carbon length 2 and 4 and 6. So they do not need activation. Similarly, medium chain fatty acids from 4 to 8, they also do not need activation. Whereas long chain fatty acids more than 14 carbon length, they need activations. So this short and medium chain fatty acids, they directly enter from the cytoplasm into the mitochondria for beta oxidation. Whereas the long chain fatty acids, they undergo activation with the help of the enzyme thiokinase or fatty acyl CoA synthetase. As the name synthetase states that it requires energy from ATP hydrolysis, and this ATP is hydrolyzed to AMP and inorganic pyrophosphate, which drives the reaction forward and activates the fatty acids to long chain fatty acyl CoA. Now, this needs a special transport protein, and this transport protein is carnitine. Now, this fatty acyl CoA combines with carnitine to form fatty acyl carnitine, catalyzed by the enzyme carnitine acyl transferase. Now, so there is a special transport protein called carnitine transporter, which will transport this fatty acyl carnitine from cytoplasm to mitochondria. So, in the mitochondria, this fatty acyl carnitine, the carnitine is released back with the help of carnitine acyl transferase 2 to form fatty acyl CoA. Now, this will undergo 4 unique steps of beta oxidation to form acetyl CoA. Now, this fatty acyl CoA is acted upon by dehydrogenase enzyme, and it gets oxidized to trans-acyl CoA or alpha beta unsaturated fatty acyl CoA, and simultaneously FAD gets reduced to FADH2. In the next step, it undergoes hydration to form beta hydroxy fatty acyl CoA. Subsequently, again it undergoes oxidation to form beta keto fatty acyl CoA with simultaneous reduction of NAD to NADH and H+, and the enzyme is fatty beta hydroxy fatty acyl CoA dehydrogenase. Lastly, this beta keto fatty acyl CoA is cleaved by a thiolase enzyme to form fatty acyl CoA with carbon 2 carbon less, and these 2 carbon atoms are released as acetyl CoA. So the steps are repeated. These 4 steps are repeated until the entire fatty acid gets converted to acetyl CoA, which will subsequently enter into TCA cycle. So you can easily remember these 4 steps of beta oxidation, wherein fatty acyl CoA undergoes oxidation to form trans-acyl CoA, alpha beta unsaturated fatty acyl CoA, enzyme is dehydrogenase. Next step, it undergoes hydration to form beta hydroxy fatty acyl CoA. Subsequently, again it undergoes oxidation with the help of dehydrogenase enzyme to form beta keto fatty acyl CoA, and this gets cleaved by thiolase enzyme to form fatty acyl CoA with 2 carbon less. So when these steps are subsequently repeated in a sequential manner to form acetyl CoA. So you need to know what is this carnitine. This carnitine is formed from amino acid, lysine and methionine. Most commonly, this will be asked as a viva question, and it is synthesized in liver and kidney and stored in muscle. So this was all about it. Now, to make you understand in a simplified way, so I am representing this activated fatty acyl CoA with the help of a structure. You need not write the structure. If you write this much, that is more than sufficient. So, if you want to score very good marks, you can write this structure. So, here this is the fatty acid without activation. So, when it is activated, so CoA has to be added here. Okay. So this is the alpha carbon atom and beta carbon atom. So this undergoes oxidation. The first step is oxidation of fatty acyl CoA to alpha beta unsaturated fatty acyl CoA. So between the alpha and beta carbon atom, so double bond is generated here, and the compound is known as transacyl CoA, or you can, if you can easily remember, you can write this also, alpha beta unsaturated fatty acyl CoA. So in the next step, it undergoes hydration to form beta hydroxy. So at beta position, so hydroxyl group is there. So it is beta hydroxy fatty acyl CoA. In the next step, again it undergoes oxidation to form beta keto fatty acyl CoA. So these two hydrogen atoms are donated to NAD, which gets reduced to NADH. So it becomes beta keto fatty acyl CoA. So the next step, thiolase cleaves this to form fatty acyl CoA with two carbon less compound, and these two carbon compound is released as acetyl CoA. Because the oxidation involves the beta carbon atom, it is called as beta oxidation. So you can write the structures, or without structure also you can. Now coming to the energetics part, now the most common fatty acid energetic that will be asked is palmitic acid. As you all know, it is a 16 carbon compound. So this will undergo 7 cycles of beta oxidation. So how to write the overall equation is palmitic acid, because first step is dehydrogenation, which requires FAD. Next is hydration, so it requires water. The next step is again dehydrogenation, which requires NAD. So palmitic acid plus 7 FAD plus 7 H2O plus 7 NAD, and for activation it requires CoA, active form of pantothenic acid. So 7 CoA-SH. So what will be the products? What are the products that are formed here? 8 acetyl CoA plus 7 FADH2 and 7 NADH plus 7 H plus. So 8 acetyl CoA when it enters TCA cycle, it generates 10 ATP. 7 FADH2, each FADH2 generates 1.5 ATP. So 7 FADH2 generates 10.5 ATP, and 7 NADH when they enter into ETC, each NADH generates 2.5, 7 into 2.5, 17.5. So 8 acetyl CoA generates 80 ATP, 7 FADH2 generates 10.5 ATP, and 7 NADH generates 17.5 ATP. So the total number of ATP generated is 108 ATP. But there is the two ATPs are used for initial activation of the fatty acid. So the net energy yield is 106 ATP when palmitic acid is oxidized by beta oxidation. So the most common fatty acid energetic that is asked is palmitic acid. So you can remember. Now coming to the regulation part, so there are two levels of regulation. One is the allosteric regulation, and the other one is hormonal regulation. Now the hormonal regulation is done by glucagon and insulin. So glucagon is catabolic hormone and insulin is anabolic hormone. And insulin is active in well-fed state and glucagon is active in starvation state. You can remember like that. Now, insulin is, as you all know, it is an anabolic hormone. So, whenever there is increased release of insulin, it will facilitate lipogenesis, synthesis of fatty acids. So, when fatty acid synthesis is taking place, simultaneously oxidation will not take place. So, insulin will inhibit this beta oxidation. On the other hand, glucagon. So, glucagon is active in whenever we are fasting and whenever we are starving. So this glucagon stimulates lipolysis, that is mainly breakdown of triglycerides from the adipose tissue. So this triglycerides are broken down to free fatty acids. So there is influx of free fatty acids into mitochondria, which will enter into beta oxidation to provide energy when we are fasting and starving. So this is about the hormonal regulation. The other regulation is the allosteric regulation. Now, the first step in beta oxidation is activation of fatty acids to fatty acyl CoA, followed by transportation of activated fatty acids from cytoplasm to mitochondria with the help of carnitine, and the enzyme in carnitine acyltransferase 1. Now, this enzyme is inhibited by malonyl CoA. Now, what is this malonyl CoA? This malonyl CoA is an intermediate compound in the synthesis of fatty acids. So the synthesis of fatty acids starts from condensation of acetyl CoA with malonyl CoA. So the acetyl CoA undergoes carboxylation to form malonyl CoA. So whenever there is the fatty acid synthesis is taking place, simultaneously oxidation will not take place to prevent the butyl cycle. So this malonyl CoA will inhibit carnitine acyl transferase 1 mainly to prevent the butyl cycle. So these two are the two methods of regulation of beta oxidation. Coming to disorders associated with beta oxidation. So there can be deficiency of carnitine acyl transferase 1. So this enzyme is mainly present in liver. So there can be deficiency of translocase enzyme which is involved in transport of long chain fatty acyl carnitine from cytoplasm to mitochondria, or there can be deficiency of carnitine acyl transferase 2 which is mainly present in skeletal muscle. So whether it is deficiency of carnitine acyl transferase 1 or 2 or translocase, what happens? There occurs defective oxidation of all the long chain fatty acids from more than 14 carbon atom. So what happens? Because the fatty acids are not getting oxidized, so more and more glucose will be oxidized in that particular individual, resulting in hypoglycemia. So one more disorder associated with this one is deficiency of this medium chain fatty acyl CoA dehydrogenase enzyme. So what happens is actually this short chain and medium chain fatty acid do not require activation and they are directly entering from the cytoplasm into the mitochondria for oxidation. So the steps remain same, whether the short chain, medium chain, long chain, the steps remain same. The first step is oxidation with the help of FAD. Next is hydration, then again oxidation with the help of NAD and the cleavage by thiolase enzyme. So in some individuals, this enzyme can be deficient. FAD dependent dehydrogenation, oxidation of medium chain fatty acyl CoA enzyme, fatty acyl CoA when it is deficient. So what happens is now this intermediate is getting accumulated. Medium chain fatty acyl CoA gets accumulated, and this can inhibit the intermediates of gluconeogenesis and glycogenolysis. So the end product of gluconeogenesis and glycogenolysis is glucose. When these two pathways are inhibited, and the individual suffers from hypoglycemia. So only treatment aspect is increased intake of carbohydrate, and these individuals need to avoid fasting. So these are the disorders. Just to summarize the disorders. So there can be deficiency of carnitine acyl transferase 1, transferase 2, or deficiency of translocase. In all the three conditions, there occurs defective oxidation of long chain fatty acids. So more and more glucose gets oxidized, and because glucose is not available, the individual can suffer from hypoglycemia. Now, the other condition is deficiency of medium chain fatty acyl CoA dehydrogenase, and this inhibits the intermediates of gluconeogenesis and glycogenolysis. As you all know, the end product of gluconeogenesis and glycogenolysis is glucose. So here also the individual can suffer from. Okay, next question. So the most common question that will be asked is describe alpha oxidation, explain its associated disorder. Now this alpha oxidation occurs in endoplasmic reticulum and it involves microsomes. When compared to beta oxidation, this does not require prior activation, and when compared to beta oxidation which produces 106 ATP when palmitic acid oxidized, this alpha oxidation does not produce energy, and so no CoA SH is required. CoA SH is active form of pantothenic acid which is required for activation of long chain fatty acids in beta oxidation. So there is no requirement of CoA SH in alpha oxidation. Now why it is called as alpha oxidation is because it involves the alpha carbon atom when compared to beta oxidation where the oxidation starts from beta carbon atom. Now this alpha oxidation is undergone by a methylated fatty acid called phytanic acid. At beta carbon atom, the methyl group is present, so this will block beta oxidation, and that's the reason why why the oxidation starts from alpha carbon atom. So this many a time this is asked as a reasoning based question. Now these are the steps of alpha oxidation. Very simple steps are there. Now this is the phytanic acid, and it has got at beta position methyl group which will block beta oxidation. That's the reason the oxidation starts from this alpha carbon atom, and the enzyme is alpha hydroxylase. So this results in the formation of a compound called CHOH. Now this undergoes oxidation with simultaneous reduction of NAD to NADH catalyzed by the enzyme dehydrogenase, result in the
formation of a keto group at alpha position of the carbon atom. So in subsequently this undergoes decarboxylation with the help of decarboxylase enzyme, resulting in the formation of a fatty acid with one carbon less. In beta carbon, it results in the formation of a fatty acid with two carbon less. In alpha oxidation, it results in the oxidation of a carbon atom or oxidation of the fatty acid occurring at alpha carbon atom, and the end product is less by one carbon atom. Now, what is the disorder associated with this alpha oxidation is Refsum's disease. This is the alpha carbon atom. Okay, when this alpha hydroxylase enzyme is deficient, it results in a condition called Refsum's disease. So these are the salient features of Refsum's disease. It is characterized by deficiency of alpha hydroxylase. So it results in accumulation of phytanic acid. Phytanic acid is the fatty acid which has got methyl group at the beta position, and this milk contains large amount of phytanic acid, and these children should avoid consumption of milk. And the characteristic feature of Refsum's disease include retinitis pigmentosa, where there occurs degeneration of the retina, and this will hamper the visual capacity of the child. And it also results in ataxia, where characterized by loss of muscle control and coordination, and also polyneuropathy, where multiple nerves are malfunctioning. So this is polyneuropathy. So these are the three salient features of Refsum's disease: retinitis pigmentosa, ataxia, and polyneuropathy. So this was about alpha. Coming to omega oxidation. Now this omega oxidation, so the fatty acid can be numbered either from the COH end or from the methyl end. So when we number it from the methyl end, so we can number it as omega 1, omega 2 and omega 3. Now this omega oxidation is mainly done by monooxygenase enzyme. So where it requires molecular oxygen and also NADPH. So NADPH gets oxidized to NADP, and this omega carbon atom gets converted to hydroxymethyl part. Now this subsequently undergoes oxidation to form COH, and the enzyme is dehydrogenase. Simultaneously there is reduction of NAD to NADH. So the omega oxidation, the end product is formation of dicarboxylic acids. Now this is a very minor pathway. Now this occurs in endoplasmic reticulum, and it ends up with formation of dicarboxylic acid. Now this omega oxidation significance is that it operates when beta oxidation is defective. So the question that can be asked with respect to this omega oxidation is, describe omega oxidation, what is its significance? Significance you need to write. So this pathway comes into picture only when beta oxidation is defective. So coming to one more mechanism of oxidation of fatty acid is, now this is refers to oxidation of odd chain fatty acid. The question can be asked is, describe oxidation of odd chain fatty acid, explain the disorders associated with them. So whatever is the odd chain fatty acid means they will have 3 carbon, 5 carbon, 7 carbon, 9 carbon, 11 carbon and so on. All these they undergo a initial beta oxidation, and they end up with formation of propionyl CoA. Now from here the pathway is common to all of the odd chain fatty acids. Propionyl CoA undergoes carboxylation, and biotin acts as a coenzyme. Here the carbon dioxide group is transferred with the help of biotin, resulting in the formation of D-methyl CoA. Now this undergoes isomerization catalyzed by the enzyme racemase to form L-methyl CoA. This gets converted to succinyl CoA with the help of mutase enzyme, and this requires vitamin B12 or cobalamin as coenzyme. Now this succinyl CoA will enter into TCA cycle. Now as you know, in TCA cycle succinyl CoA get converted to succinate. This gets converted to fumarate, which gets oxidized to malate. Malate gets oxidized to oxaloacetate, and oxaloacetate undergoes again carboxylation to form pyruvate, and pyruvate by gluconeogenesis can get converted to glucose. Now only a part of the fatty acid that is gluconeogenic is propionyl CoA. Now this is again one of the reasoning based question asked. Why is propionyl CoA gluconeogenic substrate? So you need to write this part, and the reason is, now so this TCA cycle is mainly concerned with oxidation of acetyl CoA. Now this acetyl CoA in the TCA cycle get converted to citrate, citrate gets converted to isocitrate. Isocitrate undergoes oxidative decarboxylation to form alpha ketoglutarate, and the first carbon atom of acetyl CoA gets released as carbon dioxide. Now, in the subsequent step, alpha ketoglutarate undergoes oxidative decarboxylation to form succinyl CoA. Here, the second carbon atom of acetyl CoA gets converted to is released as carbon dioxide. Now, so succinyl CoA is entering into a TCA cycle where both the where acetyl CoA is completely oxidized. Now, that's the reason why propionyl CoA is gluconeogenic. So, you need to write this part of the pathway also and this part of the pathway also. Coming to the disorders, when propionyl CoA carboxylase enzyme is deficient, it results in a condition called propionic acidemia. And when methylmalonic aciduria results when there is deficiency of racemase and when there is deficiency of mutase enzyme. So it results in a condition called methylmalonic aciduria. So many of these patients responds to B12 because B12 acts as a coenzyme for mutase enzyme. So for individuals suffering with methylmalonic aciduria, supplementation in B12 provides some amount of improvement. So this was about oxidation of odd chain fatty acids. Coming to one more type of oxidation of fatty acids. Now this mainly involves very long chain fatty acids, carbon length more than 22. Now this does not take place in mitochondria. It is mainly taking place in peroxisomes. So the very long chain fatty acids with the carbon length more than 22 and go directly to peroxisomes. Now here they undergo activation with the help of fatty acyl CoA synthetase enzyme or thiokinase to form very long chain fatty acyl CoA. Now this very long chain fatty acyl CoA gets oxidized to alpha beta unsaturated very long chain fatty acyl CoA or trans NOL CoA, and the enzyme is FAD dependent dehydrogenase. Now let's compare and contrast this with beta oxidation. What's the difference between peroxisomal oxidation of very long chain fatty acids and beta oxidation of long chain fatty acids? See, just check out the first step is very much similar, but the in peroxisome what happens, this FADH2 that is formed during the first step of the reaction, so it donates the hydrogen atom to oxygen and this gets converted to hydrogen peroxide. Now this hydrogen peroxide is utilized to engulf all the pathogens that enter inside the cell. So excess hydrogen peroxide that is generated in this reaction is acted upon by catalase and it is safely reduced back to water. When compared to beta oxidation, now this is just an oxidation reaction where fatty acyl CoA gets oxidized to alpha beta unsaturated fatty acyl CoA or trans NOL CoA. In peroxisome, there is generation of hydrogen peroxide that you need to remember. Remaining steps are similar to beta oxidation. So in the next step, alpha beta unsaturated very long chain fatty acyl CoA undergoes hydration to form beta hydroxy very long chain fatty acyl CoA. This undergoes oxidation with the help of dehydrogenase enzyme where NAD gets reduced to NADH and beta hydroxy very long chain fatty acyl CoA gets converted to beta keto very long chain fatty acyl CoA. Here the this product is cleaved by thiolase enzyme to form very long chain fatty acyl CoA minus 2 carbon atom, and these 2 carbon atoms form acetyl CoA which will enter into TCA cycle. Now this sequential oxidation takes place until the fatty acid gets converted to a 8 carbon fatty acid. From here, from the peroxisome, now this 8 carbon length long chain fatty acid goes to mitochondria and the beta oxidation continues. So this is how you need to write the flow chart for peroxisomal oxidation of fatty acids, which mainly involves very long chain fatty acids with carbon length more than 20 or 22. Okay, so the characteristic feature of this peroxisomal oxidation is generation of hydrogen peroxide, which is utilized by the cells to engulf all the pathogen. Now, so next coming to oxidation of unsaturated fatty acid. What do you understand by unsaturated fatty acid? So those fatty acids which have double bond in their structure. So here I am describing the oxidation of monounsaturated fatty acid, 18 carbon compound, that is oleic acid. Now this oleic acid has got a double bond at position 9 and this is cis in nature. Okay, now during oxidation of this monounsaturated fatty acids, first because it is 18 carbon compound and it has got a double bond at position 9, it will undergo 3 cycles of beta oxidation. We know the end product of beta oxidation is acetyl CoA. Here 3 cycles of beta oxidation means 3 molecules of acetyl CoA are released, and each molecule of acetyl CoA has got 2 carbons, so totally 6 carbons will be released here. So 18 minus 6 is 12 carbon. So now it will undergo 3 cycles of beta oxidation, and the beta oxidation continues unless and until it reaches the double bond. Now this is double bond fatty acids are naturally occurring fatty acids, but metabolism involves trans double bond. So there occurs isomerization from cis to trans double bond. Okay, so now the enzyme is able to act here. Now once the cis to trans transformation takes place, now this becomes alpha beta unsaturated fatty acyl CoA. Okay, check out. So this is alpha carbon atom, this is a beta carbon atom. So this gets it gets converted to alpha beta unsaturated fatty acyl CoA, also called as trans NOIL CoA. So the first step of the beta oxidation gets bypassed and it directly forms alpha beta unsaturated fatty acyl CoA. This undergoes hydration to form beta keto fatty acyl CoA. This gets oxidized by NAD dependent dehydrogenase to form beta keto fatty acyl CoA, which is cleaved by thiolase enzyme to form fatty acyl CoA with 2 carbon atoms less, and these 2 carbon atoms are released as acetyl CoA, which will enter into TCA cycle. So only difference you need to remember between this and the beta oxidation is, so the beta oxidation initially starts until it encounters the double bond. So there is change in double bond from cis to trans because metabolism involves only the trans double bond fatty acids. So rest all remains, the step remains same. So the energy yield varies because here the first step of beta oxidation characterized by oxidation of the fatty acid to form alpha beta unsaturated fatty acid using FAD dependent dehydrogenase is, that step is lacking. So the energy yield will be less by 1.5 ATP per double bond. So this sometimes this is asked as a Viva question, which you remember. Okay, so this was all about oxidation of various types of fatty acids. Coming to the self-assessment part, to know how would you understand. The major pathway for fatty acid oxidation is beta oxidation, okay, which takes place in mitochondria. And the most common fatty acid that undergoes oxidation is saturated fatty acid. The energetics that will be asked is palmitic acid, which you need to remember. Beta oxidation of fatty acid occurs in mitochondria. The rate limiting step of beta oxidation involves carnitine chatter. So this holds good mainly for long chain fatty acids, not for medium and short chain fatty acids, which directly enter from the cytoplasm into the mitochondria. Only long chain fatty acids carbon length more than 14 and 16 require carnitine shuttle for them to be transported from cytoplasm to mitochondria. Carnitine is required for transport of only long chain fatty acid with carbon length more than 14 carbon. The first step in beta oxidation is oxidation, FAD dependent dehydrogenase reaction. Each cycle of beta oxidation produces what? It produces both FADH2 and NADH and H+, so which are channelized into ETC and they generate. Alpha oxidation is required for the metabolism of branch chain fatty acid, mainly the phytanic acid, which is present in the classic substrate for alpha oxidation is phytanic acid, which is present in milk, and hence these children should avoid consumption of. Defect in alpha oxidation leads to which disease? Refsum's disease, characterized by retinitis pigmentosa, where the retinal degeneration occurs and vision is affected. Ataxia, cerebral ataxia, the coordination of the muscles and gait is altered, and also polyneuropathy, malfunctioning of multiple nerves inside the child's. Omega oxidation occurs in endoplasmic reticulum. The key enzyme in omega oxidation is cytochrome P450. It's a monooxygenase. Omega oxidation produces what? Dicarboxylic acids. Okay, so omega, because it involves a omega carbon atom which gets hydroxylated to form CHOH and gets oxidized to form COH, so it results in the formation of carboxylic acids. Increased omega oxidation is seen in, so only when beta oxidation is defective, omega oxidation will come into picture and it will result in the formation of carboxylic acids. Medium chain fatty acyl CoA dehydrogenase deficiency leads to what? Decreased fatty acid oxidation. Now we know that medium chain fatty acids directly enter from the cytoplasm to mitochondria for oxidation, and there are 4 steps in the oxidation of fatty acids in the mitochondria, which involves oxidation. First FAD dependent oxidation, then hydration, then NAD dependent oxidation, and then the cleavage. So here what happens? Here the FAD dependent and the NAD dependent oxidation of fatty acid is not taking place, so fatty acids are not oxidized, and what happens, inside the child utilizes whatever the carbohydrates the child is consuming, so the glucose gets oxidized to provide energy. When glucose is exhausted, they tend to suffer from hypoglycemia. So this was all about oxidation of fatty acids. So to get a comprehensive picture of the lipid metabolism, please do watch all the lipid metabolism videos that will be posted sequentially over a period of 2 weeks to get the comprehensive picture and for having complete clarity. So the next video will be on denosynthesis of fatty acids and synthesis of triglycerides. Thank you. Please do like, share, and subscribe. Do check for your MCQ score. If it is the score is more than 50%, it's very good. Less than 50%, do re-watch the video and please do retake the MCQ test. Thank you. Happy learning.